So a large portion of this should be review, and if it is not review from your freshman year, you need to be sure to be putting in the time to make sure that you know this. These are things that you are expected to know. They are prior knowledge, and their assumptions will be made on your test that these are a non-issue, easy things for you. Okay, so let's start with the concept of cell theory. Okay, cell theory basically has three parts: that all living things are going to be made up of cells. All cells will come from pre-existing cells, and that cells are the basic units of life. Okay? In that sense, you cannot break a cell down any further than it already is. Okay? If you try to take this DNA out of this prokaryotic cell, or start taking out these mitochondria, this Golgi apparatus, these things cannot live by themselves. So cells are our basic units of life. And cells do not just appear out of thin air. Okay, they have to come from a pre-existing cell, as a general rule from mitosis, or with a prokaryotic cell from binary fission. So let's take a second then and look at how a cell is limited. And size is a huge issue for cells. Okay, so cell size is going to be limited, and one of the factors that's going to be an issue here is that the surface area of a volume, the surface area of a sphere or a cube, is going to increase slower than the volume. And this is important because the cell membrane, if y'all remember, the cell membrane is what regulates what goes in and out of a cell. And so the cell membrane is only on the surface area. And if the cell has a really large volume, then there's not enough surface area to bring in the nutrients that the cell needs. And so cells are going to be three-dimensional objects. Okay? They're mostly round in your animal cells. You've got a variety of shapes with your prokaryotic cells. But all the nutrition, all their nutrients has to come across that cell membrane. And the larger the cell is, the longer it's going to take for things to diffuse to the inside. Okay? So let's say I have a cell that is, if I have a cell that's this big, I can't draw, okay? And let's say it takes 10 seconds for things to diffuse all the way to the center. Then I have a cell that's this big, okay? And now it's going to take like 20 seconds for things to diffuse to the center. So the larger the cell is, the longer it takes the materials to diffuse to the center. And that could become an issue if the cell isn't getting its nutrients fast enough. So we can calculate this with our surface area to volume ratio. Okay, so you see on this first little cube here, okay, I'm going to take my surface area, six-sided cube, each are just a unit of 1 there. Okay, so my surface area is 6, my volume is 1, so my surface to volume ratio is a 6. Okay, now, when I blow this cell up in size, okay, and so now I've blown it up in size, and it's at height now of 5. Okay, so I've got the squares of 5, so my surface area is 150. My total volume, height times width times length, okay, is 125, so now my surface area to volume ratio is 1.2. Okay, so my surface area to volume ratio has decreased significantly. Okay, but if I take this large cell and I divide it into a bunch of smaller cells, so I still have the same volume. My volume remained constant, okay, but I increased my surface area. By chopping it up into all these other little cells, I have increased my surface area now. Now my surface area is 750. Now I get back to that surface area to volume ratio of a 6, which would be much more efficient for the cell. So remember, if y'all remember back to your freshman year, that's one of the reasons why a cell would go through mitosis, is if it got too large. And that's because it's trying to do this. It needs to bring its surface area to volume ratio. It needs to increase that. It needs to increase the amount of surface area to the amount of volume that is there. Hopefully the plan is that we will be able to do a lab where y'all can actually see this happening. So y'all should hopefully remember your two basic kinds of cells. Remember, we've got a prokaryotic cell versus a eukaryotic cell. So what we have here, hopefully you recognize as prokaryotes. That's an A. Okay? And so your prokaryotes are going to be your bacterial cells. And they are the oldest. They were the first to evolve. Okay? And they've got a variety of characteristics, and we'll kind of walk through some of these. Okay? So most of your prokaryotic or bacterial cells will have a cell wall. Not necessarily all of them. And some of them will be made out of what's called peptidoglycan. There'll be a variety of things. You'll have some bacterial cells that may be made out of lipids, some that are made out of uric acid. It's just it's a variety. But a lot of your bacterial cells will have a cell wall. That cell wall helps keep the bacterial cell from swelling and bursting. And we've got 3 basic shapes. Ooh, Apple decided to spell that for me. Okay, let me fix that spelling. Okay, we've got 3 basic shapes of our bacterial cells. Okay, we've got coccus, which is round. It won't. Okay, trying again, now that it's fixed. Okay, so we have coccus, which is a circular or round shaped. Bacillus, which would be an elongated, more rod shape. That's what you see up here. Okay, and then spirilla, which would be a spiral shaped. Okay, so those are our 3 basic bacterial shapes, and obviously the cell wall would have an impact on which shape it would be. Some of your bacterial cells will have a capsule. Okay, that capsule would be on the outside of the cell wall. If you look very closely here, you can see where that's been marked out. Okay, so the capsule will be on the outside of the cell wall, and that's going to help it adhere to other substrates, other organisms. It can offer additional protection, and again, not all of them will have this capsule. They will all have to have a plasma membrane. Remember, a plasma membrane is your cell membrane. Okay, and remember, the main job of your plasma or cell membrane is to control what goes in and out of the cell. Okay, it helps maintain homeostasis by regulating what can go in and out of the cell. And that's the same for a prokaryote as it is for a eukaryote. Okay, your bacterial cells will also have a nucleoid region. Okay, it does not have a nucleus. Remember, there's no membrane-bound organelles, but it does have an area of the cell where the DNA will kind of congregate and clump together, and that would be the nucleoid region. Okay, and so those would be some of your prokaryotic characteristics. So let's look at a different picture of a prokaryote. So I've still got this prokaryote here, okay, and you can see here I've got my capsule, cell wall, plasma membrane. Okay, capsule, if it has a capsule on the outside, cell wall, then the plasma membrane. Okay, there's the nucleoid region with that basically circular DNA there. All right, and then let's look at some of the other things that are here in this bacterial cell. All right, it's got ribosomes. Remember what ribosomes do? Ribosomes do protein synthesis. All organisms, doesn't matter prokaryotic, eukaryotic, have to be able to do protein synthesis. They have to have enzymes. So we've got ribosomes there. Some of your bacterial cells will have a flagellum. Most of you know it as flagella, which is plural. You know, the flagellum will help propel or completely move the cell from one place to another. Okay, so some of them will have a flagellum. Most of them will have pili, which are these little things here, these little spikes that are sticking up all over it. Okay, those pili are important for DNA exchange. You know, bacteria essentially reproduce by cloning, by binary fission. But you remember, based on evolution, it's very important that we get variation within the organism. And so one of the ways that bacteria can get variation in their DNA is when they come in contact with other bacteria, these pili will allow them to stick to one another and exchange parts of DNA. So even though they're not doing sexual reproduction, they can still exchange DNA when those pili come together and attach them to other bacterial cells. Some of your prokaryotic cells will have a lamella. Okay, and the lamella is important in your photosynthetic prokaryotic cells. The ones that do photosynthesis have to have somewhere to have these pigments, and they have to have some kind of membrane. And so the lamella is the pigment where these photosynthetic pigments would be. And then most of your bacterial cells have what are called plasmids. Okay, so there's your spelling there, your plasmid. Plasmids will be circular pieces of DNA. They are not part of the genome, so they're not part of the bacterial chromosome. They'll just be this little circular piece of DNA, but they do contain genes. This is how, and when we talk about biotechnology, this will make a lot more sense. But this is how we do a lot of that synthetic production of, let's say, insulin. You're trying to make other cells produce insulin so we can make insulin for diabetics. Okay? We take the gene for insulin production and we insert it in a plasmid, so then the bacterial cell will start making insulin. So plasmid is where we do a lot of gene manipulation when we're talking about biotechnology. Your bacterial cells have a wide, they've got a wide range of how they can get their nutrition. Okay? You can have decomposers or pathogenic bacteria, both of which would be heterotrophs, right? Okay, so these would be heterotrophs. Whoa, that's terrible writing. So decomposers would break down your dead things. Remember, your heterotroph gets its nutrition from an outside source. It does not produce its own food. So your heterotrophs there, okay, would be your decomposers or your pathogenic bacteria. Remember, pathogenic makes you sick. And then your photosynthetic and your chemosynthetic, those would be autotrophs. Remember, autotrophs make their own food. Okay, and so you have either light, so they do photosynthesis and they convert the light energy to chemical energy, or chemosynthetic, where essentially they'll break down chemicals. Okay, like these things can break down rock, they could break down iron, and they will convert that into their nutrition. And they can also do a wide variety of biochemical pathways. Even though they don't have membrane-bound organelles, some of them can still do, obviously they can do photosynthesis if they're an autotroph, photosynthetic autotroph. Some of them are what are called obligative anaerobes. Obligate anaerobes do not use any oxygen at all. They are able to produce ATP without oxygen, and for some of them, even the presence of oxygen will even kill them. And you've got some that are facultative anaerobes. Facultative anaerobes can switch back and forth. If oxygen is present, they can do aerobic respiration, but if it is not, they are capable of switching back to do anaerobic respiration. And then you have some bacterial or prokaryotes that are able to do cellular respiration. Now that we've looked at prokaryotic cells, let's take a brief minute to look at eukaryotic cells here. Okay, so you remember your big difference between the two is that eukaryotic cells are going to have membrane-bound organelles. Okay, and remember we've got that endosymbiotic theory. Okay, if you guys remember that from evolution. Let's see if we can make this fit. Okay, so that endosymbiosis or endosymbiotic theory, things like the mitochondria, the chloroplast, how those things came from ancestral prokaryotes. You see here you've got the original prokaryote, okay, and we have the invagination, the infolding here of the cell membrane, and that's what's going to start forming these membrane-bound organelles. Okay, and then getting these, here's your aerobic heterotrophic prokaryote that's engulfed by the larger one, as well as the photosynthetic. And over time, okay, their symbiotic relationship becomes so close that they can no longer live separately. And so our eukaryotic organisms, again, they're going to have these membrane-bound organelles, which allow for the division of labor. Okay, and those membrane-bound organelles, some of them are going to have thought to have come from that endosymbiotic theory. Okay, some more support for endosymbiosis is that these organisms, or these organelles here, the mitochondrion, our chloroplast, those are going to have characteristics that support this endosymbiotic theory. Like they have their own circular DNA, which is very similar to a prokaryotic DNA. They're able to replicate like a prokaryote. Okay. They are also have their ribosomes are similar, as well as how they do protein synthesis are all similar to prokaryotic cells. So both plant and animal cells would be eukaryotic cells. Remember how to spell that. Remember, if you have trouble, remember you have eukaryotic cells. Some people still mix up prokaryote versus eukaryote. So you have eukaryotic cells. You have cells that have membrane-bound organelles. Okay, so let's look here. This is our animal cell. Hopefully you are able to recognize that. Our animal cell over here, our plant cell over here. Okay, remember in plant versus animal cell, we've got a few things that are going to be a little bit different. Okay, so animal cells will not have a chloroplast. They don't need one. They don't do photosynthesis. No central vacuole, but they do have other vacuoles and vesicles, but not a central vacuole. In the tonoplast is the membrane. You can see it over here. The tonoplast is the membrane that surrounds the central vacuole. So obviously an animal cell wouldn't have a tonoplast. No cell wall and no plasmodesmata. And we're going to talk about what a plasmodesmata is near the end of this, but that's essentially a way that plant cells can communicate with one another. Plant cells, on the other hand, are going to be missing some things that animal cells have. Okay, down here you'll see that your plant cells aren't going to have lysosomes. They do digestive properties. They're not going to have centrioles. They're not going to have flagella, with the exception of some plant sperm. In particular, in our